Hi guys, and welcome to today's episode of Biology. In today's episode, we are going to be focusing on testing foods. In this video, we are going to look at the 4 different tests on food, namely testing foods for starch, reducing sugars, fats, and proteins. We can test for the presence of starch in foods using iodine solution. The test is done by simply adding iodine solution to a sample of the food. If there is starch present, a blue-black color is obtained. If there is no starch, the iodine solution remains orange-brown. On the other hand, we can test for the presence of reducing sugars in foods by adding Benedict solution to a food and heating it. The steps include cutting or grinding a little of the food into very small pieces. Put these into a test tube. Add some water and shake it up to try to dissolve it. Add some Benedict solution. Heat the tube to 80 degrees Celsius in a water bath. If there is reducing sugar present in the food, a brick red precipitate will form. This test works because the reducing sugar reduces the blue copper salts in Benedict solution to a red compound. However, if there is only small traces of reducing sugar in the food sample, the solution would turn green or yellow after heating. Meanwhile, the solution would turn orange-red after heating if there is a moderate amount of reducing sugars. Fats can be tested using the ethanol emulsion test. Firstly, chop the food and shake it up with ethanol. Although fats will not dissolve in water, they do dissolve in ethanol. Next, pour the ethanol into another tube containing distilled water. Do not pour any of the solid food pieces from the first tube. A milky appearance shows that there is fat in the food. The fat ethanol mixture breaks up into millions of tiny droplets when it is mixed with the water. This mixture is called an emulsion. If there was no fat in the food, the mixture of water and ethanol remains transparent. The test for proteins is called the burette test. This is done by mixing the food in water and adding some potassium hydroxide solution. Then add 2 drops of copper sulfate solution. Shake the tube gently. If a purple color appears, then protein is present. If there is no protein, the mixture stays blue. This test can also be done by using a ready-mix reagent called burette reagent, which contains both potassium hydroxide and copper sulfate solution already mixed together. In summary, the test for starch includes the use of iodine solution. A positive result will be a blue-black color. The test for reducing sugars involves the use of Benedict solution and heating the sample in a water bath to 80 degrees Celsius. A positive result would show a brick red precipitate. Meanwhile, the test for fats includes ethanol and water, while the positive result would be a milky or cloudy appearance. Lastly, the test for proteins involves a mixture of potassium hydroxide solution and copper sulfate solution or burette reagent, and a positive result would be a purple colored solution. Hope this video helps. Thank you for watching. Bye.